let me welcome you cordially to the session. As time is flying, we are going to start, despite plenty of people still are sheathing themselves. Okay. I can't help. Okay, so let me introduce the team of pharmacologists from the Moravia, which is part of the Czech Republic, the central city, Brno. And we are working at Masaryk University, which is situated there. And for a long time, as you will see, we are dedicated to research, pharmacological, preclinical, especially research, in the field of cannabinoids and endocannabinoids. Despite in the program, there is my name without any title of the talk, I am going to ask you, kindly don't mind that I'm having a talk too, and this will be a historical overview of our results in the field of research of cannabinoids at our department. Okay, can I have, yeah, slides is there. So we also have been astonished about the nice, sorry, herb cannabis, and we've been since the beginning aware that this is a mixture of plenty of substances which could be biologically active and plenty of cannabinoids as well. And we know at the beginning of our starting to study cannabinoids that pharmacological mechanism of action was not clear, and how it was, why we do have these days here. Because as you can remember, in 1964, there was isolated in its pure form THC, and this was something. Professor Meshulam then structurally established this substance, which was for pharmacologists a big deal, because having such a clear substance, well established, they can start with pharmacological experimentals. Okay, this is how from that year a number of citations regarding cannabinoid and cannabis research was rapidly increasing and still is increasing. You can check it on the website. So how it is with the pharmacological mechanism of cannabinoid and the history? In 1964, as I said, we didn't know anything, which was bad for pharmacologists. In 1988, there was verified specific binding sites for THC, and those were called, according to pharmacological tendency, the receptors, cannabinoid receptors, in a shortish CB receptors. In 1992, there was isolated an endogenous ligand of these binding sites, and you know that this was anandamide, and this was the first when Moravian people come to the discovery of endocannabinoid system, because Dr. Lumír Hanuš is a doctor from Palacký University Olomouc, which is another town of Moravia, and he was dedicated to chemical investigation of cannabis as a young person and for a long time, and then when he came to work with Professor Meshulam, he was that one who isolated from the pig brains endogenous cannabinoid titled anandamide, as you know, all of you perhaps. And cannabinoid receptors were then very verified because their CB receptors were cloned, firstly at rats, then in humans, and very recently after that, there was discovered that perhaps this is not just a simply one cannabinoid receptor type, at least two CB1 called and CB2 receptors, both coupled to G proteins, and their activation inhibits adenyl cyclase and thus inhibit conversion of AMP to cyclic AMP. However, at the beginning it was thought that CB2 are distributed just in periphery and CB1 in brain, which was changed afterwards, and further studies confirmed other receptors which are ready to bind endocannabinoid as well as exogenic cannabinoids, like these vanilloid receptors, and there are some others which are called orphan G-brain coupled receptors, and endogenous cannabinoids didn't stay just on anandamide, and this was CB1, CB2 receptor agonist as well as vanilloid receptor agonist. And then again, Dr. Hanuš come with 2-HG, which is also very similar to anandamide binding sites, and then come in 2000 just vanilloid receptor agonist and arachidonally dopamine, and then again Hanuš come with noladin, CB1 agonist and partial agonist for vanilloid receptors, and what is important, nowadays we do have also virodamin, which is CB1 receptor either antagonist, full antagonist, or at least partial agonist, and CB2 receptor agonist. This endogenous cannabinoid is much more higher in concentration in peripheral tissues than in periphery. So we can conclude that at present we can recognize these types of cannabinoids, organic ones from cannabis plants and endocannabinoids from our body and other vertebrates' bodies. And however, so we do have these subtypes, and what was not mentioned that much here yet, we do have also plenty of synthetic cannabinoids of a variety of distinct chemical classes which are acting at cannabinoid receptors differentially as a selective or non-selective agonist or partial agonist or antagonist or even inverse agonist. So this was present here firstly on this conference by Professor Meshulam, that on the central nervous system, function of endocannabinoids at synapses can be explained like this. After release of neurotransmitter and binding to postsynaptic receptors, the membrane of postsynaptic neuronal membrane is reacting by synthesis of endocannabinoids, which are released and bound to specific presynaptic level cannabinoid receptors, or they are also the rest of which is not bound, transported by transport into the neuronal cell, and there they are very fast biodegraded. And it was dedicated that the endocannabinoid system looks like to be a part of protection against various damages in the central nervous system overactivity. However, we have to be aware that its hyperactivity could be dangerous to us as well. So, for better therapy, better science is needed. I do hope that you agree with that sentence. And it was said already in this year or published that historically effective therapeutic treatments have been developed for diseases only if adequate animal models were available. And this is a principle of translational medicine which we are using, which we would love to use in our experimentations of our team, because this is an association of experimental and clinical research methods. Okay, so when there is a phenotype of disease, then should be stated the animal model, and then biomarkers in that model should be stated, and then there can be found pathways and networks, and these 3 points are the points which we are following in our research. Okay? We started very fast after the discovery of endocannabinoid system and reports of Dr. Hanush to us, we start to collaborate with the Hebrew University labs of Professor Meshulam, and this was the first publication which was very well accepted by the scientific community, and we've shown that on the effects of aggressivity in mice, cannabinoids, agonists like anandamide, endogenous one, and synthetic UJ210 supplied by Hebrew University have a biphasic effects, differential at small and large doses, increase in aggressivity after small doses in submissive animals, and decrease after large doses in dominant aggressive animals. Then also in leukocyte phagocytosis as one of cell immune activity, we have found a biphasic effects with the same agents, same cannabinoids, after small doses increase, after large doses decrease of this leukocyte phagocytic activity. And this was published and well accepted, and as you can check on the screen now, this was confirmed by others in differential experiments till these days. So I think this is very important to bear in mind that cannabinoids can work that way biphasically. There started to be taking care about that there could be a competition between cannabinoid effects and calcium levels in cells, and thus we decided to put on possible functional competition in simultaneously given anandamide and calcium channel blocker verapamil, and we've been inspired by this here mentioned publication. However, we've been oriented towards central nervous system effects and also the immune function of leukocyte phagocytosis. And you can see that anandamide and verapamil interact together on the agonistic behavior, as here is saline administration, verapamil administration, no changes of aggressivity of our mice on PERT interactions, and anandamide suppressed that effect. However, when combined with verapamil, this effect disappeared. Okay, then on leukocyte phagocytosis, it was very similar because the red wine is a control. Anandamide at that dose increase of activity. However, after putting together with verapamil, green, it was suppressed, that effect, as well as a high dose of 10 milligrams per kilogram, where if this is a red control, suppression is a anandamide effect, and the yellow one when the verapamil was added and that effect was disappeared. Okay. I'm afraid that at that time when I've been sitting in the audience, there was no experimental results dealing with antagonists of CB receptors, and this is a rimonabant which was mentioned marginally here, and it was very important too for discoveries of physiological and pathophysiological acts of endocannabinoid system, because it was a wonderful tool, pharmacological tool for experimentation like that. And this is our model of drug addiction, IV drug self-administration of drugs in rats, and methamphetamine was in our interest, and you can see that if the active whole, when is used by the animal, there is put an injection of the drug into the circulation of the animal, and very easily that response to the active whole with the hut including in it is increasing the intake of the drug. And this is how it was with changes of this intake of methamphetamine in rats by administration of anandamide as a pretreatment. And you can see that it looks like slightly there was an increase of intake of methamphetamine. Then we used a methanandamide supplied by Hebrew University, and this increase was even larger, but still not significant. Methanandamide is a metabolically more stable agent. However, what was very important, that antagonists of CB1 receptors, synthetic agent AM215, which is by some researchers also characterized as an inverse agonist even, at all doses used as a premedication, suppressed intake of methamphetamine. That's why my question was if for the drug treatment of addiction would be nice to have available the antagonists at CB1 receptors. Okay, these our publications were very well accepted, as you can see on the screen. And further, some others continue with the same attitude and receive the same results, even in the 2011 when they copied our experimental design. However, they put the cannabinoid antagonist intracerebrally right into the reward pathway, into the intraaccumbens. This was also said here several times that cannabinoid receptors play important role in regulation of food intake, energetic balance, and lipid and glucose metabolism. That means that this can influence cardiometabolic risk factors, called also by internal medicine people as a metabolic syndrome, and this was studied, and the implication of CB1 receptor activation was confirmed at the central nervous system level and also peripheral tissue level, as you can see on the screen easily by yourself, because it's quite big, that slide. And what we did, very simple experiment, but the results are as we expected. CB1 receptor antagonist, AM215, which is a synthetic one, at a dose given for 8 days repeatedly, suppressed the weight of animals, and this was without changes of their hypermotility. So this was very significant result. So sometimes cannabis effects are called as a gateway for usage of hard drug of use, and some studies are reporting that it is like that, some are not, and we've been interested if effects of cannabinoids can be somehow associated with the phenomenon which is called behavior sensitization, because behavior sensitization is believed and is confirmed to increase wanting and craving in addicts, and this is because at nucleus accumbens level, this is a relation among neuronal plasticity and behavior changes leading to addiction, that way that there is increase of compulsive drug seeking and drug taking behavior. Behavior sensitization was described very well by Robinson and Berridge from Cambridge, England, as a phenomenon occurring during repeated intake of drugs of abuse. And what is also important, that afterwards there was confirmed existence of cross-sensitization, and behavior sensitization causes a very progressive and long-term increase in behavior drug effects. and those neuroplastic changes I have mentioned, confirmed in striatum, contribute to compulsive drug-seeking and drug-taking behavior, which plays an important role for development of addiction. However, it's believed that suppression of behavior sensitization could be suggested as one of possible approaches how to treat addiction. Okay, very easy way how to check development and expression of behavior sensitization in rodents is to check the locomotor activity after amphetamines, which are stimulating also, of course, the activity. And you can see here if this is a trajectories of naive mice, and after one dose of methamphetamine, they is increased. However, when the drug is given simultaneously, repeatedly, the sensitizing effect is apparent as a much more increased locomotor activity. And these are our experiments when we sensitized mice to methamphetamine by administration of CB1 receptor agonist methanandamide. And you can see, you can see that this is a classical sensitization with repeated doses of methamphetamine, and this is the same or even more stimulated animals after sensitization to methamphetamine challenge dose by that cannabinoid. And on the opposite, how worked CB1 antagonist there? This is a classical sensitizing, and even after the washout period of 7 days still was there, increased stimulation after the methamphetamine challenge dose. And when there was a premedication with CB1 antagonist, this disappeared and we received non-significant changes. Okay. And also CB2 receptors, synthetic 1 GVJH015, produce the similar effects of suppression of sensitization. Okay. This is methamphetamine. Dose of that CB2 receptor antagonist produce no changes, however, suppressed sensitization to methamphetamine. We've been also interested how it is with the expression of CB1 receptors, and we use the real-time PCR for measuring of that. And as you can check on the slides after the sensitizing experiment, cannabinoid increase the CB1 receptor expression, as well as its state after the administration of methamphetamine, while methamphetamine didn't change it. Okay. Then cannabidiol was mentioned several times in differential talks here, and we know it's called a non-psychoactive cannabinoid, and this is the history, and very much has been done by Professor Pertwee in Aberdeen, and since those years mentioned here, the strategy to explore cannabinoid receptor agonists as medicine was put with the GW pharmaceuticals. This slide was also presented here already, and we can see that cannabidiol nowadays is experimentally in interest for plenty or most than the others indications as a potential drug. And we work with cannabidiol in the social defeat model in mice as a model when the repeated social defeat can produce the depressive behavior in the open field, and we can see, or you can see, that this is the suppression of behavior in the open field after the repeated social defeat, and that was neglected after the administration of cannabidiol. And another model, a rat model of depression, well known and well validated, is olfactory bulbectomy, and the same results we have received here because the cannabidiol suppressed the effects of olfactory bulbectomy in rats. And these are some other data published about possibility that cannabidiol is acting as an antidepressant, which also can add to treatment of addiction, because on the withdrawal, patients are depressed, or even there exists very often a comorbidity of depression and addiction. And what are pharmacological strategies for therapeutic use of cannabinoids? It's somehow completely different to usage of cannabis as a herbal treatment, because pharmacologists, they are trying to identify compounds that retain therapeutic effects without side effects, simple compounds, single compounds. And pharmacologists are searching for alternative methods of delivery, and they are searching for usage of synthetic cannabinoids, very well defined, which will be very selective ligands of individual cannabinoid receptors, or even nowadays pharmacologists are also searching for possibility how to manipulate with the activity of the endocannabinoid system, that way that the drug will be changing synthesis or degradation of endocannabinoids, or will be a ligand of selective transporter for the reuptake of the endocannabinoids, and such ways. We are here from the one team, which is a research group of experimental applied neuropsychopharmacology, and we are oriented towards experimental pharmacology on the behavior research. We are using animal models of anxiety, aggressivity, depression, schizophrenia, addiction, and Parkinson's disease at present. And this is just one insight into the laboratory. And another part of our team is interested especially in the pharmacokinetic research, and especially on biotransformation of psychotropics by cytochrome P450 enzymes and their interaction and gender differences, and they are using isolated perfused rat liver, intracellular drug administration of rats, and pharmacokinetic experimental studies in rats in vivo. And this is again insight into the lab. And this you can, I'm going to leave it for a short time on a screen. You can leave it our more recent publications dealing with the endocannabinoid system research, as well as most recent. And we do hope to continue such a research and publications of our results. And I thank you for your attention. And now we are going to the program you have in printed version. And I'm going to invite Vincenzo Micale, which is our Italian colleague working with us for a longer time already. And for his talk, The Endogenous Cannabinoid System as a Potential Therapeutic Target in Schizophrenia, Preclinical and Clinical Evidence.
Thank you, Alexandra. So good morning, everybody. Thanks for the kind invitation. So today I will show some data which support the idea that the modulation of the endocannabinoid system could be a potential therapeutical target to treat schizoaffective disorders. Maybe no. So So, schizophrenia is a psychiatric disorder affecting 1% of the population worldwide, and it is characterized by three main class of symptoms. The positive symptoms, hallucinations and delusions, the negative symptoms, social withdrawal and anhedonia, and the cognitive deficit. We still don't know exactly the pathophysiology of schizophrenia. Different hypotheses have been done. The oldest hypothesis is that in the schizophrenia is related to the overactivation of the dopaminergic system in the mesolimbic pathway, which induce the positive symptoms, the psychosis. Other hypothesis is that the NMDA receptor hypothesis, so there is a hypofunction of NMDA glutamate transmission system, which mediates the negative, the positive, and the cognitive symptoms. More recently, hypotheses are based on the fact that schizophrenia is a kind of a neurodegenerative disorders where prenatal anoxia or infection, excitotoxicity could facilitate the development of the disease. Or neurodevelopmental hypothesis, based on the fact that the exposure of the neonat during the gestation or during the first period of the life to some adverse environmental stimulus could increase the risk of to develop the disease. As you know about the pharmacological treatment, we have two main classes of compounds. We have the typical antipsychotics and the atypical antipsychotics. Typical, which preferentially blocks D2 receptors, atypical block D2 receptors and serotonin receptors as well. But as you can see, these both class of compound induce different and several side effects, and there are several patients which don't respond to the pharmacological treatment. So it's necessary to improve the treatment, especially for the negative and the cognitive deficit, which are not treated, which are not reversed by typical and atypical antipsychotics. So, a lot of data suggests that the endocannabinoid system could be involved in the pathogenesis of the disease, and the modulation of this system could be a potential therapeutic target. So, endocannabinoid system is a neuromodulatory system. It is characterized by the two main endogenous cannabinoids, anandamide and 2-AG, the enzymes for the synthesis and the degradation, FAH and MAGL, and the receptors CB1, CB2, and the vanilloid receptors. So, lot of data suggests that the endocannabinoid system is involved in the several physiological processes. So, this regulation of these systems seems to be involved in a different disease, as neurodegenerative disorders, cardiovascular disease, obesity, multiple sclerosis, mood disorders, and so. So, there are a lot of clinical data suggests the so-called cannabinoid hypothesis of schizophrenia. One of the first data is from the group of Piomelli. They show that in schizophrenic patients, they have a high level of anandamide in the cerebrospinal fluid. And high level also another endocannabinoids, endogenous cannabinoids, palmityl ethanolamide, and no difference about another endogenous cannabinoids, OEA. And the same group later showed that the schizophrenic patients which low cannabis use, they have high level of anandamide in cerebrospinal fluid, but Another group of schizophrenic patients which are characterized by high cannabis use, they don't have. So there is a reduction of anandamide level after frequently cannabis use. Other data suggesting the dysregulation of endocannabinoid system in the schizophrenia. This, again, the group of Daniele Piomelli. So Andrea Giuffrida, what they shown that in the schizophrenic patients, again, there is a high level of anandamide in cerebrospinal fluid, and this high level of anandamide is not counteracted by atypical antipsychotics, but is reversed by typical antipsychotics. And it means that this high level of anandamide is specific of schizophrenia, because in patients with dementia or affective disorders, there was not any difference as compared to the healthy, to the control. And in the same study, they found a negative correlation between a high level of anandamide and psychosis. Other clinical studies, they found a high level of the group of Coete, they found a high level of anandamide in the initial prodromal state of psychosis. It means that anandamide, anandamide is more specific. And a recent study showed that QAG seems to be involved in the schizophrenia as well, because they found that in the brain, in the hippocampus and in prefrontal cortex, they found a high level of QAG. These are the which was reversed by antipsychotic treatment. Again, as for anandamide, the antipsychotic treatment was able to reverse the high level of endogenous cannabinoids. Other data suggest that there is also a dysregulation of cannabinoid receptors. So, for instance, it has been seen an increasing of the binding of CB1 receptor in schizophrenia patients in different brain regions, as in dorsolateral prefrontal cortex, anterior singular cortex, and the posterior singular cortex. In a recent paper, it has been seen that it seems that the CB2 receptors are involved in the schizophrenia, because it has been seen that in patients with in the schizophrenic patients there is low expression of CB2 receptors in the total lymphocytes T and in the lymphocytes T helper. So, but from the title of the talk, so I'm going to show clinical data and the preclinical data. So it means the preclinical data, they came from animal models of schizophrenia. So what does animal models means? So animal models is an experimental setup which try to mimic the disease in terms of symptoms or behavioral signs, biological mechanism underlying the disease, and to try to mimic to see the efficacy of the pharmacological treatment. So it means that a good animal model of schizophrenia, but in general mood disorders, has to show face validity, construct validity, and predictive validity. About the face validity in animal models of schizophrenia. So in humans, we know we have the positive, negative, and the cognitive deficit and the sensory motor gating deficit. So in animal models of schizophrenia, the positive deficit are mimicked by the increase of locomotor activity. The negative symptoms in humans, as social withdrawal and anhedonia, in the animals, we could assess the social behavior in rats using the social interaction test and so. Or also evaluating the immobility in the forced swim or the nest building behavior. The deficit, the cognitive deficit in humans, we have different tasks in animals to evaluate the cognitive impairments. And the sensory motor getting deficit in humans, we could evaluate through the evaluation of the prepulse inhibition of startle response. This is one of the most translational tests which we have. So, animal models of schizophrenia. Based on the hypothesis of schizophrenia, we have different animal models. We have the pharmacological models, so based on the treatment with dopaminergic agonists or NMDA antagonists. We have the lesions models, the environmental neurodevelopmental models, based on the isolation of prematernal malnutrition, of the exposure to MAM. And also we have genetic models through the use of the knockout animals. So we have three different ways to evaluate the effects of the modulation of endocannabinoid system in the animal models. One of the first ways through the direct activation of CB1 and CB2 receptors. So in this case, we could use CB1, CB2 agonists, or selective CB1 or CB2 agonists. Or we could attenuate the endocannabinoid signaling through the use of CB1 and the CB2 antagonists, or we could test the knockout animals. Or we could facilitate the endocannabinoid signaling through the use of the blockers of the degradation, so inhibitors of the enzyme for degradation, or we could block the transport of the anandamide. So, let's go to see the effects of the pharmacological modulation, the effects of some compounds which modulate endocannabinoid system on positive symptoms. So, what it has been seen that CB1, CB2 agonists, like WIN, CP, were able to decrease the hyperlocomotor activity in different animal models of schizophrenia. Almost they are pharmacological models. And the CB1 antagonists, inverse agonists, like Rimonabant, was able to reduce the hyperlocomotion as well in different animal models, in different pharmacological models of schizophrenia. About the effects of the modulation of endocannabinoid system on the negative symptoms, again, the CB1 and CB2 agonists were able to decrease the social impairment in pharmacological models. And again, the CB1 antagonist, inverse agonist, was able to reduce the social impairment in the pharmacological models. About the effects of these compounds in the cognitive deficit, here we saw that almost the activation of CB1 and CB2 receptor worsens the cognitive deficit. There is an increase of the cognitive deficit or the disruption of the prepulse inhibition. By contrast, the antagonist, the CB1 antagonist, was able to reduce the cognitive deficit or the deficit in the prepulse inhibition. But the problem of these two classes of compounds, we cannot use these two classes of compounds, CB1 and CB2 agonists or CB1 antagonist, inverse agonist. CB1 and CB2 agonists we cannot use for the psychotropic side effects. The CB1 antagonist, Rimonabant, although there were nice results, nice preclinical results, we cannot use Rimonabant because, as you know, Rimonabant was withdrawn from the market due to the psychiatric side effects. So, And also there were not strong positive clinical data about the effects of rimonabant in the schizophrenia. So there was just recently there was a decrease of psychosis. But anyway, we cannot use rimonabant or CB1 antagonist inverse agonists. So, promises compound for the treatment of schizophrenia is cannabidiol. Cannabidiol is the major non-psychotropic cannabinoid, so different from the CB1 and CB2 agonists. And cannabidiol, it seems that it works, it has several mechanisms of action. So it's able to inhibit the degradation and the uptake of anandamide. So there is an increase of anandamide levels. It's able to stimulate TRPV1 receptors and is a serotonin 5-HT1 receptor agonist. It's able to inhibit the uptake of the adenosine. About the effects of cannabidiol in the preclinical models of schizophrenia, it has been seen that it was able to decrease the hyperlocomotor effects induced by amphetamine and ketamine, and was able to reduce the cognitive deficit, again in the model, in the cognitive deficit induced by THC of the NMDA antagonist. So these are preclinical data. Really recent, one more preclinical data. This was published by the group of Francisco Guimaraes 1 month ago. What they saw, that cannabidiol was able to revert the PPI deficit induced by NMDA antagonists, was able to revert the social deficit induced again by the NMDA antagonists, and also the cognitive deficit induced by NMDA antagonists. It means that at the molecular level was able to revert the decrease of parvalbumin, which is an index of GABAergic functionality. About the clinical studies. So, the first clinical study in 1995 showed that the cannabidiol was able to reduce the psychotic symptoms. Another study, again, cannabidiol was able to reduce psychotic symptoms like amisulpride. Another study, and so several studies, clinical studies suggest the antipsychotic effects of cannabidiol. This is the most recent study, again from the group from Leveke. What they found, that cannabidiol was able to reduce the psychosis as amisulpride, and it seems that the effects of cannabidiol is related to the increase of anandamide level. So this is related to the mechanism of action of cannabidiol, which blocks the degradation of anandamide and blocks the reuptake. And cannabidiol didn't induce the side effects as amisulpride, like the extrapyramidal side effects and the increase of the weight gain. So, it's important because, as I showed in one of the previous slides, one of the problems of the antipsychotics treatment is the side effects. So, the hypothesis behind the effects of cannabidiol, so it means that cannabidiol blocking the degradation of anandamide and blocking the anandamide reuptake. So after cannabidiol treatment there is a high level of anandamide, which, stimulating the CB1 receptors, block the dopaminergic activity. So, from this data, we could conclude that, of course, there is a dysregulation of endocannabinoid system in terms of endogenous cannabinoids or receptor in the clinical, in the humans, and it means also in the experimental models of the disease. So the modulation, since there are this dysregulation, the modulation of endocannabinoid tone could be a good option for treatment of schizophrenia. But we cannot use the CB1 antagonist, inverse agonist for the side effects. So at least one idea could be to use the neutral antagonist, the CB1 neutral antagonist. And from the clinical data and from the preclinical data, cannabidiol seems to be the most promised compound for to treat the symptoms of schizophrenia. So in the last, I have 1 minute? Okay. Just, I will show some preclinical data from our lab. So we are working on a neurodevelopmental model of schizophrenia induced by methylazoxymethanol. What we found? We found that we treated the pregnant rats at gestational day 17, and already in the neonatal age, the pups, they have deficit. They have a kind of deficit, as we can see from a delayed appearance of a neonatal reflex. So it means that there is a kind of problem in the brain maturation. And these, at the neonatal age, these animals, they show a high level of endocannabinoids in the male, 2 Ag, and no significant of anandamide. And the changes in endocannabinoid system is stronger in the female. High level of anandamide, 2 Ag, and decrease of expression of enzymes of degradation. These animals at adolescent age, they shown deficit in the social behavior, male and female as well, and partial deficit, partial cognitive deficit, because we tested these animals in the novel object recognition test, male and female. So these data support that in these animal models of schizophrenia, there is alteration of endocannabinoid system, which already at neonatal age. Thanks for your attention. Thanks. Thanks all the people which are involved.
Thank you very much, Vincenzo. And I'm going to ask for the audience for a favor to postpone the discussion to the end of the session, if you kindly don't mind. And I'm asking Jana Kučerová for her presentation according to printed materials you have in your program.
So thank you very much for inviting me. Thank you very much for your attention that you're keeping from the very beginning of the morning. And I would like to share with you just a short story from our experiment. So it's real data from our lab. Unlike my colleagues, I don't have any overview of the current knowledge. So this is, I will try to make it focused, short, and fun. So what we are generally studying is the self-medication hypothesis, which is a theory which says that in depression, the people tend to self-administer, tend to be more prone to develop drug addiction, any drug addiction, including cannabinoid addiction or cannabis addiction. So there is a clinically well-established, well-known comorbidity of drug addiction and depression, which could be, well, the hypothesis could be obviously two, at least. It depends which is first. Either depression, which then tends to be self-medicated by a drug of abuse, Or first could happen the drug addiction, and then the depressive phenotype could follow. This is especially true for psychostimulants, where it's obvious, but both hypotheses are probably true, and it depends on a specific population of the patients. What we are operating is the comorbidity of depression and drug addiction on the base of the self-medication of the depressive state and the potential monoaminergic deficit from the very old theory of the development of depression. And we are trying to model this in animal models, in rat models. So we have developed a rat model of comorbid depression and methamphetamine addiction already, where we have shown that the depressive-like rats do self-administer, so do voluntarily take more drug, more methamphetamine than the sham-operated controls. I will later show you the method. So the aim of this particular study was to translate this model for cannabinoid addiction and to see whether, because methamphetamine and cannabinoids have very different mechanism of action, so to see whether this disturbance or this increase in the drug addiction, this high tendency, is linked to the depressive phenotype or specific rather for methamphetamine, or it could be translated to cannabinoids as well. We were using WIN. I will just refer WIN 552122, so I will just refer to this as WIN, which you probably know. And this is a dose per infusion, so that the animal could actually get more infusions in one session. And we were using a special kind of rats. It's called Lister-hooded rat. Lister-hooded rats are usually used for any studies with cannabinoids because they seem to be more vulnerable or more sensitive to any cannabinoid action. So the methods we are using, it's already mentioned olfactory bulbectomy as a well-established model of depression. It's a surgical model which consists in a removal of the olfactory bulbs. I'm sorry, right here. And it actually leads to very similar symptoms of a human major depression. So there is a behavioral syndrome, there is a behavioral phenotype, very specific. There is a catecholinergic deficit, serotonergic deficit specifically. Also neuroendocrine dysregulation, they have higher corticosteroid levels. They have immunodeficits, so the phenotype is pretty similar to what we see in people. For the drug abuse, we used operant IV self-administration paradigm, which is based on the original idea of Skinner's idea from the behaviorist group, in which the animal is trained to voluntarily self-administer the drug in an operand box. So using some kind of operandum, a lever, or in our case, a nose poke. And after a stimulation of this operandum, an intravenous infusion is delivered by the infusion pump. These are our operandums. This is the nose poke, which is equipped with a photo beam. When the animal puts the head in it, the photo beam is broken, and it makes a signal after which the drug is delivered. So the animal learns to self-administer the drug quite soon. Starts just by exploring the environment, starts to see that it could be a pleasant feeling after stimulation of the specific operandum. So this is how the method works. Furthermore, we wanted to see what's happening in the reward pathway, because we are studying mostly drug addiction. This is the primary aim. So what's happening in the crucial part of the reward pathway in the brain of the rat, so nucleus accumbens, we employed so-called in vivo microdialysis, which consists in a surgery again. We put a special probe to the brain of the animal, which is which is done under general anesthesia, obviously, which has an inlet and an outlet. And here you can see a detail of a semi-permeable membrane. So when we then perfuse after short recovery, we start the session, and we perfuse the probe with a solution, take the samples and analyze the samples. So we have in vivo data on the extracellular levels of neurotransmitters in I wouldn't say real time, but every 20 minutes. So we can evaluate the effect of a drug on this neurotransmitter release. So the protocol of these studies, there are two studies. First one on IV self-administration. So the first what we had to do was the olfactory bulbectomy surgery to develop the phenotype. Then we performed the IV self-administration surgery, so which consists in implantation of the intravenous catheter. We established the behavior profile of the OB animals before we started, and then we performed the IV self-administration part, which started with acquisition, so that where animal starts to understand what's happening and can start to self-administer the drug. And then, as a model of treatment, let's say discontinuation of the drug to the drug, we employed a method which is called extinction, when in the operant cage the drug is replaced with vehicle, saline in our case, and so the animal sees or feels that there is no way how to get those pleasant feelings in that operant box, and starts to extinguish the drug-seeking behavior, so stimulation of the operandi. And the microdialysis study was just after the recovery from OB surgery in a different batch of animals. We started the microdialysis session, started taking the basal samples, and then we injected the animals 1 dose of WIN to see what is the response in the nucleus accumbens of these animals. So let me convince you a little bit that this model of depressive-like phenotype really works and could be translated to human medicine. This is sucrose preference test, so it's a measure of anhedonia, which is typical for depressive people. You can see that in sham-operated, so the control animals, which are the white columns, they tend to self-administer when they can choose actually out of 2 bottles, one with 2% sugar solution and the other one with water. They drink 90% of the intake of the water. They take the sweet one, as opposed to the OB rats, which take something about 55% after 4 hours, and even a little bit less after 24 hours. So this is the phenotype that we expected. Another behavior characteristics of the OB model is the hyperlocomotion after exposure to novel environment. So we put the animals to locomotor cage, to the activity cage, which automatically measures the locomotion. And you can see that after 5 minutes, the horizontal activity of the OB rats, which was much higher, just like the vertical activity. It's not as significant. Vertical activity means that the animal is standing up, is rearing. This gets normalized at the time point of 10 minutes, because the environment is no longer novel. So this is the dysregulation, this is the maladaptation phase of the behavior. And this is already a first result from the self-administration study. You can see that it was in 30 days, so a long study, where every day the animal was introduced to the operant cage and was able to self-administer the drug. And the black dots are OB animals, the white line is the control animals, and what we see here is actually the active the stimulations of the active operandum, or as well the number of infusions, actually, because it's 1 to 1. So after 9 days, 9th day, it was the first day when the 2 groups actually started to be very dissimilar, and at the second half, let's say second second 2 weeks of the experiment, you can see that the intake was very stable and very different in the two groups, with the OB animals self-administering almost a double of the sham-operated controls. If you're interested in the dose, then it's roughly 250 micrograms of WIN per kilogram and 500 micrograms of half a milligram of WIN in OB animals. This is the latency to take the first dose. When the animal is introduced to the cage, we measure the time to the first infusion. And so you can see that again the OB animals actually want the drug or take the drug starting from roughly the day 10. After shorter time, or start to self-administer the drug in the cage after shorter time than the sham-operated controls throughout the study. It could be interesting to see whether the intake has some kind of pattern, whether the OB rats, for instance, would have taken the drug at the beginning as opposed to the sham at the end. This is not the case. The intake is completely regular. Every line here means one infusion. The first 3 animals are sham-operated, the second 3 animals are OB. These are typical patterns of the intake from the end of the study. And you can see that there is no specific time to take the drug. It's the self-administration happens at constant rate throughout the session, which was, by the way, 2 hours daily. Just the intake in OB animals is higher. Then we switched the animals to the extinction part of the study when the methamphetamine was substituted with saline only, so there was no reward. The animal didn't feel anything in the cage. But still, you can see that actually the so-called extinction criteria, basically that they stopped to try, were met in the OB rats after 3 weeks and something, almost 4 weeks. Their stimulation of the active operandum has dropped under 10. So they kept trying at least 10 times throughout the 2-hour session for 3 weeks, whether there will be anything or not, as opposed to sham-operated animals, which have given up after 10 days, roughly. Just to sum up the self-administration study, the OB animals seem to want more drug, want it quickly, and they give up later when they find out that there is no way how to get the drug. So we wanted to see what's happening in the brain of the animals. So this is a separate study, different animals. And we were measuring dopaminergic level of the dopamine in the nucleus accumbens in the sham-operated and OB rat, which was the same at the beginning, but this graph is in percent. So this is a percent of the release of dopamine after the WIN dose. So we administered the WIN here, as you can see, but it was 0.3 milligrams per kilogram. It's important. And you can see that in sham-operated animals, there was a typical dopaminergic release. Any reward is related to dopaminergic release in the nucleus accumbens, so we expected that. However, in the OB animals, there is nothing. And I mentioned that the dose is important. This is the dose which you might remember is typically self-administered by the sham-operated animals. So it seems, we can't be conclusive, but it seems that the sham-operated animals really do feel some reward after this dose, and this could actually explain why the OB animals tend to get at least twice as much in order to produce probably the same effect, the reward effect. So this was this was our data. We can't really draw much conclusions out of it, but we are validating a model for future drug testing. And these are acknowledgements for my colleagues for the grants. I can announce that this article was actually already published relatively recently in Frontiers in Neuropharmacology. Thank you very much for your attention.
We are thanking you too. However, we have to proceed.
Hello, good morning. My name is Juřica. I'm going to continue with a different topic concerning cannabinoids and endocannabinoids possibly. And my talk is aimed on influence of cannabinoids and endocannabinoids on the activity of drug metabolizing enzymes. When we speak about drug metabolizing enzymes, let me to let you know that the most of drug metabolizing enzymes is comprised by cytochrome P450. Cytochrome P450 is a hemoprotein which is present at most in the liver, also in the intestine, but in the brain as well. And cytochrome P450 has many isoenzymes, many subtypes of this enzyme, which is responsible for metabolizing distinct group of chemicals, including drugs, including xenobiotics. So that's for introduction of what cytochrome P450 enzymes are. Concerning interaction of endocannabinoid system or cannabinoid system, cannabinoid drugs and endocannabinoid system and drug metabolizing enzymes, there are several perspectives from which this interaction could be seen. The first is the problematic of polymorphisms, induction or inhibition of drug metabolizing enzymes by xenobiotics. Drug metabolizing enzymes, and mostly cytochrome P450, is polymorphic. That means that in the population there are several groups of metabolizers, like extensive, rapid ones, slow metabolizers, and this, of course, may affect concentration of drugs in the blood and thus affect as well. So that's one thing. If cannabinoids and endocannabinoids may affect concentration of other drugs, then because of drug metabolizing enzymes also may convert endocannabinoids and exogenous cannabinoids to active or inactive substances or active in other way. And the third perspective is how endocannabinoid system is involved or is not involved in the regulation of cytochrome P450 activity. And this may be managed by systemic effects. I mean, it means on the liver, at the site of the liver, or effects of endocannabinoid system and interconnection with other neuroregulatory systems within CNS. So the first perspective, polymorphisms, how polymorphisms and how drug metabolizing enzymes interact with cannabinoids. So metabolites of arachidonic acid, which is a precursor of many endocannabinoids. Arachidonic acid is converted via cytochrome P450 enzymes to many biologically active ligands of cannabinoid receptors, CB receptors. Anandamide, as several times mentioned, endocannabinoid, is the substrate of many cytochrome P450, apart from fatty acids, amide hydrolase enzymes as well. But anandamide is converted via several isoenzymes towards low affinity ligands, CB ligands, cannabinoid ligands, and via different cytochrome P450, it means 3A4 isoenzyme towards higher affinity and stable, more stable cannabinoid ligands of CB2 receptors. And of course, polymorphisms in these enzymes may affect the activity or levels of endocannabinoids at all. So this has been in part mentioned that endocannabinoids are converted to inactive, mostly inactive metabolites by fatty acids, amide hydrolase, epoxy hydrolase, cyclooxygenase, and lipoxygenase. But by cytochrome P450 enzymes, it is converted occasionally also to more active and stable, more stable metabolites. Concerning cytochrome P450 enzymes, there are many of isoenzymes, and those which are marked with red are also present in the brain. That means that the metabolism may occur already at the site of the action, already at the target beyond blood-brain barrier, so it means in the brain. So this is, I have already mentioned that via fatty acids, amide hydrolase and epoxy hydrolase, cyclooxygenase and lipoxygenase, metabolites are less active, but through to cytochrome P450, metabolites may become even more active or more stable at least. And 41 out of totally 57 cytochrome P450 enzymes are also present in the brain, and 20 of them are present in significant amounts in several brain regions. This is a schema which may illustrate how metabolism may be complex and complicated. So, for instance, anandamide may be converted via several isoenzymes towards different metabolites. So this is for illustration how complicated it could be. And of course, different compounds, different metabolites may proceed different biological effects. And polymorphism in a single enzyme may promote different effects at all. But these changes are not too big, as I could hypothesize that these effects are not clinically relevant, as I suppose. Biotransformation of exogenous cannabinoids is also proceed by cytochrome P450, for illustration, tetrahydrocannabinol, cannabidiol, cannabinol, and synthetic compounds are made by polymorphic and not polymorphic enzymes. So this problematic may become even more complicated. Another topic that drug metabolizing enzymes may interfere with the effects of other drugs, such as paracetamol, very well-known drug, and at least some analgesic effects are produced due to interaction with the endocannabinoid system, because metabolite of paracetamol is an inhibitor of anandamide cellular uptake and is an agonist of vanilloid receptor. And by blocking CB1 receptors, analgesic properties of paracetamol are also blocked. Concerning the second perspective, how endocannabinoid system may influence cytochrome P450 enzymes, so cannabinoid non-selective agonists usually increase the activity of some isoenzymes or cytochrome P450, such as this synthetic agonist CP55940, and you can see it here. That's the activity, that's the content, and that's the specific activity of cytochrome P450. And again, it was repeated in a similar way that acute and chronic administration of anandamide increases cytochrome P450 activity of distinct isoenzymes. On the other hand, cannabidiol inhibits in vitro metabolic activity of 3A4 isoenzymes, but cannabidiol, as had been mentioned in Vincenzo's presentation, cannabidiol is lacking psychotropic effects and is not a direct agonist of or strong agonist of CB receptors, so it's somehow exclusive from the group of phytocannabinoids. And third perspective, how pharmacokinetic drug metabolizing enzymes may be regulated by means of interaction of endocannabinoid system and other neuroregulatory systems. So let me show you briefly how drug metabolizing enzymes are influenced by diet or xenobiotics. It can be direct influence, means inhibition or some cooperative allosteric modulation of metabolic activity of these enzymes. Then it could be proceed by means of interaction of glucocorticoid receptor and some other drug, some other gene gene promoters. Then, of course, peripheral hormones may influence these receptors as well. And recently it was investigated by the team of Professor Daniel in Polish Academy of Science in Krakow that dopamine, at least dopamine, norepinephrine, may influence release of pituitary hormones and thus also metabolic activity of liver cytochrome P450 enzymes. So that's the experiment they have performed at tuberoinfundibular and mesolimbic dopaminergic pathways are enrolled in the regulation of hepatic P450, and mechanical disruption increases activity of 1A and decreases activity of other isoenzymes of P450. Moreover, dopaminergic receptors are dimerized with CB1 receptors, at least in nucleus accumbens, but it appears also in some other brain regions. And CB1 receptors are also co-expressed with other types of receptors, such as serotonin receptors, some kinds of serotonin receptors. And another study confirmed that co-stimulation of both CB1 and dopaminergic receptors modulates both receptor signaling on both neuroregulatory pathways. So there are some other studies confirming that there is a link between dopaminergic and noradrenergic pathways. For instance, if dopamine release is inhibited by gamma-aminobutyric acid, by tonic inhibition of made by GABA. GABA may be excreted by interneurons, so the release of GABA blocks dopamine release. But when a cannabinoid agonist administered its WIN, similar to those which Jana, probably chemically similar to those Jana has used in her experiments. So agonism on CB1 receptor diminish release of GABA, and that's disinhibition of dopamine release. And if we block CB receptors, we block this dopamine release as well. So there is another link between these systems. So we have asked a question if the cytochrome P450 activity may be influenced by means of endocannabinoid system modulation, and we have performed by this interaction, for instance, and we have performed an experiment with Vistar rats, Vistar albino rats. And we have operated a cannula into nucleus accumbens core in a stereotactic frame. And we have, I will show you where it was. So it was nucleus accumbens core. So these are coordinates where we have introduced a guide cannula, and we have administered saline to controls and cannabinoid agonist, CB1, CB2 agonist, O2545 hydrochloride intracerebrally in 1 microliter, just 1 microliter. It was a concentration of 11 micromolar. And we have performed an experiment then after 10 days of administration with selective substrates of cytochrome P450 metabolic activity. That means that we have administered in an isolated perfused rat liver experiment. We have administered to this perfusion system diclofenac, which is a selective substrate of cytochrome P450 to C11 to C611 isoenzyme. And we have observed the velocity of the reaction of biotransformation from diclofenac towards 4-hydroxydiclofenac. And that's the measure of the metabolic activity. Higher metabolic ratio of non-metabolized to metabolized drug, high ratio is slower metabolism. So there is no difference, obviously, at this isoenzyme. There is no difference observed in a 2D2 isoenzyme, which is known to be non-inducible. But we have seen a difference in phenacetin dethylation towards paracetamol. Phenacetin is a prodrug of paracetamol, and it's converted by 1A2 isoenzyme, and we have observed an increase, it's a decrease of ratio. That means ratio of prodrug to metabolite. It means increase in drug activity of this enzyme. So it's increased metabolism. So to answer the question, if endocannabinoid system is probably involved or may be involved in cytochrome P450 regulation, so response could be yes, probably it is, but we have to perform many other studies. Well, I'd like to conclude, and I'd like to thank you for your attention, and thank to my colleagues at the department, and thank you for inviting me to this symposium. Thanks.
Thank you very much, Honza, and thank you for standing to stay with us. And we will be more than happy if we will be able to answer any of your questions, if there are some.
Thank you. I have a question to Vincenzo. It was a very nice overview and presentation. So I have one comment that you mentioned that there is no clinical evidence on the effect of CB1 antagonist on schizophrenia. This is not entirely true, because even after the withdrawal of Rimonabant, Sanofi almost for 1 year continued a study with its analog, and it was actually very effective, but finally, because of the milieu of the FDA not likely approval, they discontinued, but it was quite effective. And my second question, that it is regarding the CBD's beneficial effect on in schizophrenia through endocannabinoids. So it is quite well known that THC through CB1 receptors may exacerbate schizophrenia, and even in patients who are not, they're prone to schizophrenia, cannabis use can exacerbate it through most likely CB1. And you mentioned that CBD elevates endocannabinoids, and through CB1 it is protective. Isn't that possible that actually increased levels of endocannabinoids lead to downregulation of CB1, and that's why actually you see the effect?
Could be. So about the first question about the CB1 antagonist and the Rimonabant. So the question is, okay, the preclinical data, they show nice results. The clinical data about the Rimonabant in the schizophrenia, there was not strong results. So there were just few...
But I mentioned that there was another analog of Rimonabant that was not published, which was very effective, actually more effective than aripiprazole.
So maybe the next step could be to try to see if this CB1 neutral antagonist, because they don't have this inverse agonist effects. Maybe they could show efficacy in the schizophrenia, also in the other the disorders. For this, the first question. So the second question, yeah, because about this difference between cannabidiol and the THC, because what it has been seen that the THC reduce the anandamide. The people, the smokers, the schizophrenic people with high smokers of the THC, they have reduced the level of anandamide. So it means that the THC is down-regulated the anandamide levels, which we didn't find, is not been found after cannabidiol treatment.
Okay. Thank you.
You are.
I have a question for Jana. You were analyzing the behavior of animals incarcerated in a drug dispensing machine. The poor animals had nothing else to do there. The environment was highly unnatural. What about have you ever thought about the possibility of examining their behavior in a more natural environment, something like Alexander's rat park? Don't you think that the results may be different?
You mean more enriched environment?
More natural. I mean, the animals because running freely. Or
when it comes to natural, actually, I have seen just once really more natural environment where the animal can really hop. You know, they don't generally walk; they hop, which they cannot do in a home cage, which we provide, which are following the EU directives and everything, how experimental animals should be housed. So actually, you're right. There is many approaches towards drug addiction, and obviously, environmental enrichment has been proven to prevent development of drug addiction for sure. The box is 2 hours a day, and for the rest they are in the home cage, singly housed because they have the catheters. So you're right. They have the environment does have some detrimental effects on their, let's say, to anthropomorphize mood. But that's true. So to your question, no, and I'm afraid that I have no means to construct like real life stabling system for them, because for that you need several. You probably have something like that, don't you? You just thought about it. You know, I have seen it once. It's really, it's within the full room. There is a colony of rats, let's say 10, and you have to invest one room, a lot of money to really construct the apparatus. Have something has a wheel to run. It has several arenas, so it's a demanding system. It's not standard. It would be nice to see for sure, but it has been shown already that environmental enrichment does suppress the tendency to develop drug addiction. That's for sure. So our aim is to develop a model to test drugs. This is a simple, straightforward model which is reproducible relatively easily in a standardly equipped labs.
If there are no other questions, thank you very much again for staying with us, and I hope that something we bring to your minds about our research and results especially.